Mm-hmm.
Okay, so today we are going to see the topic testosterone, and in this topic we are going to see that how the testosterone is important and what is the objective of testosterone, right? So first, let's see that what do we mean by testosterone? Can you see my whiteboard?
Yeah.
Okay. Testosterone sometimes is also called as back cross, right? And this cross is particularly used to check the the genotype of an individual that is showing you the dominant characteristics. Like in our previous lessons, we have talked about the pea plant and the individuals that are showing the complete dominance, and one of the allele is dominant and the other allele is recessive, and the dominant allele is completely masking the effect of the recessive allele. For example, in pea plant, we've talked about that the plants could be tall or the plants could be short. Short, I told you that it's the recessive uh, trait, right? And it can only express itself when the individual will have small t, or you can say the recessive homozygous form. What do we mean by recessive? Recessive means that when there is an allele that is not, no, you can say, expressing itself when it is present in a heterozygous form with a dominant allele. Like, for example, in this heterozygous form. So, this small t is not going to express itself because this capital T will mask the effect of this small t, right? So, it can only express itself when it is present in the homozygous recessive form, right? And it is homozygous. Homozygous means that both the alleles are same, right?
Mm-hmm.
So this for any characteristic, not only the pea plant height, it can be for any individual who is showing you the complete dominance and the recessive characteristic or the recessive form that is only showing the recessive form because it has both the alleles that are homozygous, right? Now, if we talk about this individual that is showing us the tall height or the tall form, that can be homozygous tall, that can be heterozygous tall, right? Because this is complete dominance. And if I give you a pea plant, right, and I ask you, this is the pea plant, right? And if I ask you to tell me that by looking at this plant, to tell me that whether this plant has uh, a genotype uh, homozygous or heterozygous. So that would you will become confused. You will not be able to tell me that what could be the genotype of this individual, whether this
Mm-hmm.
individual is capital T, or this individual is capital T small t, because
Mm-hmm.
in both the cases the individual will show us a tall height, right? So, to
Yep.
evaluate this, the objective of this test cross is this, that if we want to evaluate or if we want to check the genotype of an individual who is showing us the dominant characteristic, right, in the phenotype, and to check that genotype, we need to do a test cross. Now, the question arises that how to do test cross, right? This is our next question. The objective is to check what is the objective? Objective of this test cross is to check the genotype of an individual showing dominant form in complete dominance. In complete dominance. Okay, I'm short of space here. Now I'm moving on to the next page, and now we are going to see that how to do a test cross, right? Now, for test cross, I will select the individual definitely who is showing me the the phenotype of tallness, because I need to check, or I need to evaluate the genotype of this individual who is showing me the phenotype. That could be because of the homozygous tall or heterozygous tall, and I'll cross it with an individual whose genotype is already known. Although we can't see it with the naked eye, but short individual is the one whose genotype I already know, because short plant is short because it has the recessive alleles in it, right? That's why this plant is short, right? So this individual I know would have small t, both the alleles present on the pair of chromosome, both the alleles would be small t. But this I don't know, and I need to know. This will be dependent, the genotype of this tall individual will be assessed by the progeny, or you can say by the offspring's phenotypes, that what results we will find after this cross fertilization, right? They will produce obviously some plants in the next generation, and if, I'm saying if, all the plants are tall, right? This means that this individual whose genotype I need to know, right? This individual will always be capital T, capital T. Now
Mm-hmm.
you will ask me that why, ma'am? Why you're saying that if all the individuals will be tall and this individual will be on the basis of this result, you will assess that the genotype of this individual will be capital T, capital T. Let's prove it with the help of a genetic diagram, right? So if this individual is capital T, this individual will produce gametes, obviously. And when this individual will produce gametes, all the gametes will be of the same type, and they all will have capital T in them, right? If I show you the probability, so then I have to make all the gametes, right? And when these gametes are formed, both the alleles get separate during meiosis, right? Because you know that these gametes are always produced as a result of meiosis, right? Meiosis is basically a kind of cell division in which the gametes are produced, and these gametes are always haploid cells. We have discussed it previously as well. I'm just revising it again. And meiosis always takes place in the reproductive organs for the production of male and female gametes. Let's suppose this tall plant is a male plant and this short plant is a female plant, and when meiosis takes place in the reproductive organs, like ovules and pollen grains are produced, and they will always produce, all the pollen grains will have a capital T in them, and all the ovules will be produced of the same type. And when they will fertilize, let's draw a Punnett square for the fertilization process. Capital T male gametes, small t female gametes, right? And then let's multiply them. This one will get multiplied with this. You will write here capital T small t, because this can fertilize with this. And when this can fertilize with this, let me change my color. Okay. Oh. So when this will fertilize with this, it will come here, capital T small t. And when this will fertilize with this, it will come here, capital T small t. So all the individuals phenotypically will be tall, 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 tall. As I told you that the phenotypes, all the individuals have this genotype, capital T small t. This is the genotype of all the offsprings,
Mm-hmm.
genotype, and the phenotype is
Tall.
phenotype is tall.
Tall.
So that's why I said that if all tall individuals will be produced, so this individuals whose genotype we wanted to know would be capital T, capital T. Let's suppose, ma'am, if the results will be opposite, if the results will not be all tall, right? So what do you say about that? Let's suppose the tall individual, when is crossed with a short individual, right? If the
Mm-hmm.
results are different, that there are some tall and there are some short individuals that are produced, right? So in this case, what do you say? If there are some
Usually,
tall and if there are some short individuals that are produced, both the forms are expressed. So what will you assess about this individual? I
mm,
would say that this individual would have capital T, small t. But ma'am, why?
mm,
Because of this short individual. Because this short individual will always produce, let's prove it with the help of a genetic diagram now, right? So, because this short individual will always have small t, and when they will produce gametes, definitely this tall individual will produce two types of gametes, capital T, small t, and this will produce all the gametes of the same type, right? And these gametes are always produced by meiosis, right? And now let's make a Punnett square to show the offspring results, right? Capital T, small t, small t, small t. This is a male individual, this is a female individual, or can be opposite. When this will fertilize with this, it will produce a capital T, small t. The phenotype will be tall. And when this will fertilize with this, small t, and the individual will be short. And when this will fertilize with this, capital T, small t, tall individual. And when this will fertilize with this, small t, that will be a short individual, right? So how many are this type? Two. How many are this type? Ratio two. So 1:1, you can say, if you simplify the ratio. So
mm,
some of them are phenotypically tall and some of them are going to be short, right? So this is how you can evaluate that what could be the genotype. But if you don't want to do any cross, you cannot assess that whether the tall individual is a homozygous tall individual or a heterozygous tall individual. Is that clear?
mm-hmm. Yep.
Is that clear? You got it? Okay, now let's move on to the book. Now they have given you some other examples, like for a gray chinchilla could have the genotype GG or capital G, small g, right? And when they will do that test cross, they will give you the findings that whether the individual, in the same way that, like I used a different example and your book has used a different example, but the results will remain same, right? For example, this plant when is showing you two characteristics, one is two forms, right? And there's a capital G and capital G, small g. The genotype of with the dominant phenotype, for example, will be this. And if you want to know that whether this is showing you a homo, the individual is having the tall individual will is having the capital G or small g, you know, or capital G, small g. So to know this, you need to cross it with the individual whose genotype is already known. That is the short individual, right? And then you get to know in the same way that, like I did it in front of you on the screen, that how to evaluate or how to check that whether the individual is homozygous tall or a heterozygous tall, right? I hope that's clear to you. Now, inshallah, uh, I will talk about some more inheritance patterns in the next lesson. Okay.